Can you remember the last time you took antibiotics when you were sick? Pretty convenient, right? Antibiotics are a crucial part of modern medicine, but not just because they help our sore throats. Antibiotics are essential to performing even the most basic surgical procedures to prevent deadly infections. But unfortunately, antibiotic-resistant superbugs have been appearing all around the world. How do bacteria become resistant to antibiotics? And more importantly, what can we do to remedy this threat to humanity?
Bacteria can replicate themselves every 4 to 20 minutes, making them the fastest reproducing organisms on the planet. Every time bacteria reproduce, they can acquire completely random mutations in their DNA that could potentially give the organism a new protein that can work to fight the antibiotic. Most antibiotics currently used are designed to directly kill bacteria by destroying cell membranes or interfering with protein synthesis. This life or death selection pressure actually promotes the evolution of antibiotic resistance. Imagine a scenario where bacteria are not being treated by an antibiotic. There may be resistant bacteria in the population, but because there is no antibiotic present, these bacteria don't have a survival advantage and they will not grow out of control. Now imagine a population of bacteria that is treated with an antibiotic. Suddenly, these resistant bacteria have a survival advantage and will thrive as the non-resistant bacteria are killed off. So what if there was a way to stop the bacteria without killing them? Bacteria have developed their own little method of communication called quorum sensing. That's right, they have conversations just like us. But what exactly do bacteria talk about? Bacterial genes code for the production of signaling molecules called autoinducers that are released into the surrounding environment. When these autoinducers reach a high density, they bind to signaling sites on the bacterium, and the autoinducer receptor complex is able to bind to the DNA promoters and activate the transcription of quorum sensing controlled genes. For bacteria, this is like their form of taking a census. Quorum sensing allows the bacteria to know that they are no longer working as an individual, they are working as a group. Bacteria will not work individually. Once they are confident that their population is high, that is when they will receive signals to release toxins that make us sick. Quorum sensing can even promote antibiotic tolerance by the production of biofilms. It allows bacteria to make fortresses encased in a film of extracellular polymeric substances. This biofilm acts as a barrier that our antibiotics have an incredibly hard time penetrating. It also provides an ideal environment for resistant bacteria to share their genes among the population through a process called horizontal gene transfer using mobile genetic elements such as plasmids or bacteriophages.
As you can see, quorum sensing is essential to bacterial function and survival. Conveniently, scientists have found quorum sensing inhibitors that can destroy the autoinducers or block the bacteria from finding them. Essentially, we are bullying the bacteria into silence by convincing them they don't have any friends. Sounds mean, but if we can figure out how to use QSIs in a targeted and accurate manner, we may have an excellent shot at solving the antibiotic resistant crisis. Bacteria aren't going to divide any slower, so we've got to be quick.